Welcome. It's really wonderful to have all of you here. My name is Yishan Zhen. I'm director of Carnegie Institution's embryology department. Our department is on Johns Hopkins campus, Homewood campus in Baltimore. Now I want to introduce Eric Marshall and Austin Hall to give some remarks.
Good evening. It's so wonderful to be here with all of you tonight. And thank you, Dr. Yishan Zheng, for co-hosting this evening here with us for our Kavli Prize lecture. So we've actually enjoyed partnering with the Carnegie Institution for Science since the beginning of the Kavli Prize, where Kavli Prize laureate evenings have been held here every year since 2008. So to get started, to introduce you to the prize partners, and the founder of the Kavli Foundation, Fred Kavli, whose vision led to the inception of the Kavli Prize, we'll show you a short video.
What if we could understand the origin of the universe? What if we could improve lives by controlling atomic structures? What if we could truly grasp the complexities of the human brain? The story behind the Kavli Prize started in the 1930s with a curious boy named Fred, growing up among the tall mountains of Eresfjord in Norway. A deep curiosity about nature and the universe stayed with Fred throughout his studies in physics and his entrepreneurial success in the United States. Until he eventually established a philanthropic foundation with the vision of advancing science for the benefit of humanity. One of the first activities of this foundation led to the inauguration of the Kavli Prizes, beginning in 2008. The prizes are awarded every 2 years in a partnership between the Kavli Foundation, the Norwegian Academy of Science and Letters, and the Norwegian Ministry of Education and Research. Each of 3 international prize awards consists of $1 million and a gold medal received in a ceremony in Oslo, presided over by a member of the Norwegian royal family. The Kavli Prize laureates are selected by the Norwegian Academy of Science and Letters, based on recommendations from prize committees, comprised of leading international scientists from 6 of the world's most renowned science societies and academies in the 3 scientific fields: astrophysics, nanoscience, and neuroscience. The biggest, the smallest, and the most complex. The Kavli Prize has 4 ultimate purposes: to recognize outstanding scientific research, honor highly creative scientists, promote public understanding of and appreciation for scientists and their work, and to foster international cooperation among scientists. Time and time again, we have seen that achieving these goals has been critical to making this world a better place. The Kavli Prize continues to be driven by the same sense of awe and curiosity felt by Fred Kavli as he grew up in the midst of nature at its most sublime, experiencing the vastness of the universe.
Thank you. So representatives of the three Kavli Prize partners are here today. We'll very briefly share what our role is in that. The Kavli Foundation awards the prize money and the gold medals to the Kavli Prize laureates, as well as publicizing the awards. Now, the Kavli Prize is just one of many programs of the Kavli Foundation. We have 20 international Kavli institutes at renowned research institutions around the world. We also convene meetings globally of scientists and those promoting public engagement with science to have the critical dialogue that we need around these topics now. So 14 years ago, Fred Kavli said, "Life as we know it today would not be possible without science. As we gain more knowledge about materials and processes in the universe that could open up benefits that we can't even imagine. But you have to be willing to fund science without knowledge of the benefits." So our speaker tonight, Dr. Emmanuel Charpentier, is a prime example of how research can open unexpected doors. She's one of the 2018 Kavli Prize winners in Nanoscience, and she's going to give a talk and then engage in conversation with Mr. Frank Sesno, who's director of the George Washington University School of Media and Public Affairs, an Emmy award-winning journalist, and an author of a book where you can learn how to ask more. You'll see him doing that in just a few minutes. And then you'll be able to engage with the laureate and Frank in conversation. But first, Secretary General Øystein Hove from the Norwegian Academy of Science and Letters will make some brief remarks.
Thank you very much. I am pleased to attend Professor Emmanuel Charpentier's lecture tonight. She shared the Kavli Nanoscience Prize in 2018 with Jennifer Doudna from Stanford and Virginijus Sisknis from Lithuania. And this is the evidence where she receives the prize from His Majesty the King in September last year. And here we have the prize winners last year with His Majesty the King, Harald V of Norway, in the City Hall of Oslo. The role of the Norwegian Academy of Science and Letters is to administer the Kavli Prize Week in Oslo, manage the Kavli Prize selection committees, approve the committee's selection of the prize winners, and announce the winners. The Norwegian Academy is happy and proud to be a Kavli Prize partner, along with the Norwegian Ministry of Education and Research and the Kavli Foundation, and through the prize work to promote the public understanding of science and appreciation for scientists and their work. The work with the Kavli Prizes continues, with the next round of awards to be announced on May 27, 2020, and celebrated in Oslo in early September next year. That is from the gala dinner during the prize awards. And here we have a little bit of commercial. Now it is my honor to introduce the Deputy Chief of the Norwegian Mission at the Norwegian Embassy here in Washington, Marius Dierdal.
Thank you, Øystein. Good evening, everyone. It's a pleasure to see so many people here. It is an honor to be here this evening and to speak on behalf of the Norwegian Embassy and then by extension on behalf of the Norwegian government. But first and foremost, it's a great pleasure to welcome all of you to this 2019 Kavli Prize Laureate lecture featuring Dr. Emmanuel Charpentier. Now, the discovery of CRISPR technology by Dr. Charpentier is one of the most significant scientific discoveries of this decade, or maybe of this century. And I as a non-scientist, even I understand that it comes with the potential to revolutionize everything from agriculture to medicine. So we're talking about a very powerful tool, a tool that can alter the very code of life itself, something that will profoundly shape our future in ways not yet fully understood. Clearly, this technology will affect us all, and which is why I think it also sparks important discussions. Last year, Dr. Charpentier's discovery of CRISPR earned her the Kavli Prize in Nanoscience, which is the Kavli Award that celebrates and recognizes scientists working on the smallest building blocks in nature. Her discovery, however, would have the largest impact. The Norwegian government highly values its collaboration with the Kavli Prizes and the three-way partnership between the Kavli Foundation, the Norwegian Ministry of Education and Research, and the Norwegian Academy of Science and Letters, as well as the long-standing and close ties between Norwegian and American scientists and academic institutions. We look forward to even more collaboration in the future. I would also like to thank the Carnegie Institution for hosting the annual Kavli Prize lectures. This is a long-standing tradition now. It's one that the embassy very much appreciates, and we look forward to this every year. So we are looking very much forward to this evening as well. The Kavli Prize Laureate lectures celebrate great minds and are an essential contributor to science, communications, and bringing science to the public by esteemed prize winners and ambassadors for science, such as Dr. Charpentier. So, Dr. Charpentier, congratulations on your discovery and on receiving the Kavli Prize. I look very much forward to being further enlightened this evening. Thank you.
It is really a true honor to introduce Dr. Emmanuel Charpentier. When many people think about bacteria, they think about disease-causing pathogens and the importance of washing our hands. But bacteria have shaped our planet's history, and they have continuously contributing to our ecosystems around the globe. They are also vital to our own health. We're now only beginning to understand how our body's microbiomes are shaping our immune system, as well as our longevity. In recent years, scientists, including our tonight's guest speaker, have revealed that bacteria's own defense system against viral invaders can be harnessed to mediate genome editing. How the CRISPR-Cas9 system moved from bacteria, where its immune role was discovered in 1987, to its implementation as a game-changing tool in biological and biomedical research, is a story with a large cast of characters. But there is no doubt that our guest tonight, Dr. Emmanuel Charpentier, was and remains a central player. It was she who deciphered the molecular mechanisms by which CRISPR-Cas9 works in bacteria. And with Jennifer Doudna's laboratory at University of California, Berkeley, she showed that how CRISPR-Cas9 could be used as a molecular scissors to edit any DNA sequences as we desire, including in humans. Dr. Charpentier is a French biochemist, microbiologist, and geneticist who earned her PhD in microbiology in 1995 at the Pasteur Institute in Paris. She then spent more than 20 years working in 5 countries, 7 cities, 10 institutions, and 13 different departments before settling in Berlin 3 years ago as a director at the Max Planck Institute for Infection Biology. Last year, She founded an independent research institute, the Max Planck Unit for Science of Pathogens, where today she's the scientific and managing director. She's also an honorary professor at Berlin's Humboldt University and co-founder of 2 companies developing CRISPR technology. A member of many national and international scientific academies, including those of Germany, Sweden, and United States. She has been awarded with a large number of very prestigious prizes. These include the Japan Prize, the Tang Prize, the Breakthrough Prize, the Leibniz Prize, and last year, an honor we celebrate tonight, the Kavli Prize in Nanoscience. Dr. Charpentier will give us an overview of her work, and then Frank Sesno will join her for a deeper discussion of the topic. Sesno is the director of the School of Media and Public Affairs at George Washington University. She's an Emmy Award, as you heard, she's an Emmy Award-winning journalist and creator of Planet Forward, a user-driven web and television project that highlights innovation's sustainability. His journalism career included more than 20 years at CNN, where he served as White House correspondent, anchor, and Washington bureau chief. Along with our co-hosts, the Kavli Foundation, the Royal Norwegian Embassy, and the Norwegian Academy of Sciences and Letters, we are thrilled to host Dr. Charpentier as our Fall Kavli Prize Laureate lecturer, and to have Frank Sesno here to help us celebrate her achievements. Ladies and gentlemen, please join me in welcoming a scientist whom Time magazine has called one of the most influential people in the world, Dr. Emmanuel Charpentier.
Thank you very much for the very kind introduction. This is for sure a great honor for me to be here with you, to explain you briefly what CRISPR-Cas9 is about and where it comes from. And it's a great honor for me to follow after my talk with a discussion with Mr. Frank Sesno. So some commercials. Surely I would like to thank the Kavli Foundation, the Norwegian Academy of Science and Letters, the Carnegie Science Institution, as well as the Norwegian Embassy and the Norwegian Ministry of Research and Education. I think it's great to have institutions that help to promote science and explain science to the public, institutions that also support the work of the scientists and that highlight the science that benefits humanity and society. But I think at the end of the day, every types of science end up to benefit humanity and society. So I'm just a scientist, and I'm going to try to simplify what CRISPR-Cas9 is about, and also to explain you some aspects about the real CRISPR-Cas9 mechanism in life. So as most of you may know, it's a transformative technology in life sciences. So a number of people refer to it as a technology that really revolutionizes the field of life sciences. So what is CRISPR-Cas9? So here it's a drawing that represents the mechanism. So you see in the middle of the slide the DNA, that is a molecule composing genes and genomes. So this is really the support of the genetic material. And CRISPR-Cas9 is a tool that allows to modify the DNA in a very precise fashion and at specific sites on the DNA. And it is composed of 2 components. One component is here represented as a pair of scissors, and the nature of these scissors is a protein that has enzymatic activity that allows the protein to cleave the DNA. And the specificity of the cleavage is brought by the second component that is of RNA nature, and this second component works like, in a way, a GPS system, allowing the protein to recognize really the sequence of DNA to be cleaved. So it works as a programmable fashion, I mean, as a programmable element of the mechanism. And so once the two-component system, so the protein Cas9, guided by this molecule of RNA, after this Cas9 protein cleaves the DNA, then cells use repair machineries to repair the break. And depending on the fashion scientists design the tool, one can introduce a number of changes, such as really replacing a gene by another gene, adding a new piece of DNA on the DNA, correcting mutations, introducing new mutations, and this occurs in a very precise manner. It is like you would use a software to edit a text, the DNA being the text, the genes being the words, the series of genes being the sentences, and the basis of the DNA being the letters that the software will edit. So it's the most precise gene editing technology that is on the market. An interesting aspect of the history of genetics, which I wanted to bring tonight, to also show you that gene editing is not new. The ability to modify genes is something that has been going on for the last 40, 50 years. So if you refer to the history of genetics, very briefly, the 19th century was really critical for establishing laws of origin of species, of segregation of alleles, and the DNA that was isolated at the end of the 19th century. And one needed to wait 80 years approximately to have in the middle of the last century the confirmation that DNA was a carrier of genetic information, and the establishment and elucidation of the structure of the double helix of DNA. And a little bit more than 50 years ago, the genetic code that was revealed, and followed by the need to understand the genetic code, meaning the functions of the genes and also the dynamic of the genomes. And for sure, to be able to answer those questions, one needed tools. And in the mid-70s, it already started at the end of 60s and followed in mid-70s, a number of enzymes were discovered actually in bacteria and viruses, and research done on bacteria and viruses allowed to really come to a number of tools that are even now still used in the molecular biology toolbox to somehow be able to modify the DNA. And this is critical in life sciences because since the DNA is a carrier of genetic information, the ability to modify DNA allows to really understand the code and to understand the functions of genes and to unravel pathways. So this was followed at the end of the 70s and 80s by the ability to sequence the DNA, so meaning to read the code of the DNA and to amplify the DNA. So for sure you know that DNA can be cloned, and so all those technologies were critical and daily used by biologists to understand mechanisms of life. As much as we could recombine the DNA and exchange genes by other genes or perform other types of modification, what was missing is really the ability to precisely edit the genes and the genomes. Having a tool that will work as a precise gene surgery tool. And you have a number of nucleases that have been developed, so these proteins that have the ability to cleave DNA, that have been developed at the end of the 90s and then more recently in 2010. Those technologies were working, but not as well as CRISPR-Cas9, and a way to engineer those tools that were a little bit cumbersome and also quite pricey. And this is what brought CRISPR-Cas9, so a tool that allows faster gene editing, less time consuming, and also an easier way to edit the genes. And this is thanks to the simplicity of the system with one protein, and that can be programmed by this RNA molecule that has just been to be designed to recognize a certain sequence of DNA of interest to be modified by Cas9. So what is for sure important for me to convey is that this technology originates from basic science. So originally, CRISPR-Cas9 is a mechanism that exists in bacteria. In bacteria, there are for sure multiple species of bacteria existing, and their life is quite very challenging always. The bacteria are susceptible to be attacked by a number of attackers, and one of the attackers are viruses of bacteria called bacteriophages. So you can see those particles that sit on a bacterial cell and ultimately will inject their DNA into the bacterial cell. And this can lead to viral DNA replication, production of viral particles that will ultimately kill the bacteria, but they can also, those viruses, bring new genetic traits to the bacteria. And as we have evolved innate and adaptive immune systems to defend themselves against bacterial and viral infections, bacteria, like any organism, have also evolved defense systems to be able to defend themselves against these attacks. You have different types of immune systems existing, and you may consider that the fights between bacteria and phages is very, very prominent. So there is always a battle going on, and bacteria have to react extremely fast. They can be killed extremely fast. So they have different ways to defend themselves, and CRISPR-Cas is one of these mechanisms existing. It's an adaptive mechanism in the sense that the system is first adapting to the phage during the first recognition step, and then establishes a kind of way to then recognize the phage upon a second infection, and ultimately can target the DNA of the phage and eliminate the DNA of the phage. So this is what is represented here on this picture. So you have a bacterial cell, and here just represented the different components of the CRISPR-Cas systems and how it works. What is fascinating with this system is, like very inherent to the microbial world, you have a lot of evolution going on. So CRISPR-Cas systems have evolved in multiple systems that use different mechanisms to, at the end of the day, target the phage DNA and make sure that the phage will not propagate any longer. So this is really a fascinating mechanism existing in bacteria. And in my lab, actually, we worked on a bacterium that is called Streptococcus pyogenes, also known as group A Streptococcus. So it's a strict human pathogen. You find this bacterium only in humans, and in principle, 30% of the human population is colonized by this bacterium without developing diseases. But in some cases, diseases can develop, such as pharyngitis, that are belonging to mild diseases caused by Streptococcus pyogenes, but can also cause very more severe diseases such as toxic shock syndrome or necrotizing fasciitis. So we were interested in actually different types of mechanisms of RNA nature that led us to actually the CRISPR-Cas9 mechanism. And I'm not going to explain you in details the different steps we had to go through to understand ultimately how the system works and to understand ultimately that this is a system composed by this protein that can be programmed by these RNA molecules to recognize a certain sequence of the phage DNA to then after lead to the cleavage of the phage DNA by the protein Cas9. And this natural system was then exploited to, just by changing the RNA molecule, offer a technology that allows, just by changing the RNA molecule, to adapt this technology to any types of cells and organisms, and being able to target any piece of DNA, virtually any piece of DNA. There are some more complexity on the technology, but in principle, a very versatile technology. What became clear when we started to work specifically on the CRISPR-Cas9 mechanism is that because of the simplicity of a two-component system and the possibility also to harness the system further to multiple versions of the technologies, that it will really be beneficial for the life science world. What was also very, very particular with this discovery is that the understanding of the molecular mechanism, let's say comparative to other types of research, was, at least in my lab when we started to work on the mechanism, first of all, knowledge was already available, so other scientists had started to really understand what CRISPR-Cas is about in bacteria and had already started to work on CRISPR-Cas9 system. But in my lab, comparative to other projects we were working on, actually, we could decipher this mechanism in a rather short period. And what was really fascinating as well is that when we published the paper in Science together with Jennifer Doudna in 2012, describing the last step of the mechanism of CRISPR-Cas9 and providing the user guidelines of how to use this mechanism as a technology to modify genes and genomes in cells and organisms, what was really very, very clear is that the scientists were in need of this technology, so they really applied very fast as soon as they saw this publication. They applied this technology for their genes and cells of interest, and this started really the field of research, that is really the applications of the technology and the development of the technology, and this field is called the Crispr Craze. And already a year after we had published our article in 2012, in the summer 2013, a journalist wrote this article for the journal Science with the CRISPR Craze, a bacterial immune system, yields a potentially revolutionary genome editing technique. So the applications were very fast, and there was really a high demand on a technology that would allow more precise gene surgery. So over the last 7 years, the technology has been largely developed. So first of all, very fast, early 2013, it was clear that the technology could allow to modify genes and genomes in plants, in human cells, in a number of model organisms that are used in the laboratories to understand mechanisms of life, such as flies or zebrafish, and also that it would allow to modify genes and genomes in fungi and bacteria. Having said this, modifications were also possible prior to CRISPR-Cas9, but again, there is more precision brought with CRISPR-Cas9. We certainly predicted that the technology will be really useful for the scientists and that it will provide means to really multiply the applications in the field of biotechnology, agriculture, and medicine. This technology will provide a number of advantages compared to the technologies that were available prior to CRISPR-Cas9. It's a cheap and easy technology. It's very efficient. It's very versatile. You have now different versions of the technologies that are on the market. It also allows multiplexing in the sense that one can modify multiple genes at once. A lot of improvements have been brought to the technology to be more specific, and specifically as well to really develop the tools that are necessary for the application of the technology, meaning how to bring the CRISPR-Cas9 genome editing tool in cells and one needs delivery systems. And a lot of research has been done on this aspect. Delivery systems are important because the system has to be brought to the cell in also a specific fashion and efficient fashion. Having said this, here is just a slide with drawings which you can enjoy, but it's just a way to remind me to tell you that the technology can not only modify genes, but also their expression. And it also allows to modify genes and genomes so that one can visualize the DNA in cells. One can study the epigenetic marks, which are important because there are a number of epigenetic phenomena that are associated to development of certain cancer diseases. And one can perform with the technology different tricks that also allow to really develop a lot all these screening methods to find new targets for therapeutics. So you have large applications of the technology in basic science, in research development, in medicine, in plant biology, in synthetic biology. So in life sciences, it's really what I like with CRISPR-Cas9. It originates from evolution of bacteria within the context of an arms race between bacteria and phages. But it allows to provide a tool that fits back into the evolution of other types of cells and organisms, in the sense that scientists have always been restricted to the types of cells they will study. And they will always mention that they work with model cells or model organisms. And now CRISPR-Cas9 has really simplified the genetics, so it allows the scientists to really work on cells that are more relevant, for example, for clinical situations, or more relevant to really issues that the society will be encountering in the future, such as climate change, so the ability really to accelerate the genetics in different types of plant species. It's important to have a precise technology to being able to perform gene editing, because it allows to really understand better the functions of genes, and specifically in the context of health and diseases. It allows to rewire biological pathways to provide better models for human diseases. With regard to medicine and health, the technology is important in indirect and also direct fashion. So the technology is currently developed as a tool, as a direct tool, to potentially cure rare genetic disorders. It's also used to engineer immune cells for the treatment of cancer immunotherapy. There is also a lot of research going on to try to figure out how CRISPR-Cas9 can be developed for antimicrobial and antiviral applications. In the field of drug development, it allows to accelerate these screening methodologies to find new targets for therapeutics, to provide better models to test the therapeutics in development. And you have also the aspect of gene drives to control disease vectors. In the field of agriculture, it allows really to boost the engineering of plant crops, but specifically to engineer plant crops in a precise fashion. An early application of CRISPR-Cas9 was with regard to phage-resistant probiotics, and also a technology that is very important for the engineering of industrial macroorganisms. So a lot of scientists are very excited by this technology, but surely there are some societal implications of this transformative technology. The good aspect, actually, at least of the CRISPR-Cas9 research from my point of view, is it's a very good example to highlight the need to pursue basic science. And I have to say that the media have covered quite well this aspect, and have communicated overall quite well to the public the applications of the technology, really the impact of what the technology has in the future of science. But also, even though there were some aspects, I have to say that maybe a bit worrying with the technology. I think the media were very good at highlighting the good aspects of the technology. It's important for us to really communicate that powerful technology can originate from science, which maybe would not be so much of interest. I always had difficulties to get funding for my research. And I have to say that the CRISPR-Cas9 technology was funded through funds that were not to ask how bacteria defend themselves against phages, because it's maybe not the pressing question. Yet, as a bacteriologist, one knows that working on this type of questions, one can develop this gene editing technology. A lot of genome editing technologies, most of them, as I said, originated from research done on bacteria and viruses. And then you have, for sure, a number of ethical considerations. So surely the ability to modify genes, specifically in human cells, bring the questions of how far one can go and how far the technology can go to not only modify the genome of somatic cells, but also the human germlines. And you have also ethical considerations with regard to the engineering of plant crops that will be commercialized, and for sure different views of how the ethics is considered in the world. But we can discuss this during the discussion. So you have a lot of tools that I expect to be developed in the future, and this will be, again, thanks to continue to really understand basic questions in microbial research. So you have new defense systems that have been discovered recently in addition to CRISPR-Cas9, so one may expect that other types of mechanisms will be exploited for increasing the tools in the genetic toolbox. Just briefly, research will not be possible without institutions and funding, some of which are represented here. One needs enthusiastic scientists, young scientists. These are really the young scientists who are doing the work in the lab, and these are the members of my lab, that have been associated to the CRISPR-Cas9 research, and truly collaboration that are also important. We live in a world where we need to answer biological questions in a multidisciplinary fashion, and we enjoy collaborating with a lot of scientists around the world. And I just want to finish, because I need to have enough time for the discussion. I just need to cite these two companies for conflict of interest, but also because I'm proud to say that I have co-founded with Rodger Novak and Sean Foy two companies, ERS Genomics and CRISPR Therapeutics. CRISPR Therapeutics focusing on developing the technology to treat human genetic disorders, and also trying to develop the technology to treat certain types of cancer. So they are doing a wonderful job. And I will finish my talk with this very nice drawing. And I would like to thank you for your attention. Okay.
Well, I'm not sure where to start. I think you said, I'm just a scientist.
Yes, indeed.
That's very much too modest of you. I would like to congratulate you on everything, and not to mention, and not the least of which is the Kavli Prize, and thank Kavli and everybody for being in this packed room this evening. It is wonderful to see you and to see science so celebrated and so listened to. So we will do that for a few minutes. I think some of you have put your questions in by Pigeonhole already. I'll be looking at those periodically and incorporating those into the conversation as we go, and then a little bit later we'll go to the microphones if we've got a few minutes for that. We've already got a lot of questions, so please feel free to weigh in. So this is amazing stuff, and in the introductions, I think you said very eloquently that this is a tool that can alter the code of life itself. And that is what this can do. It is, we are told, potentially cure genetic diseases, fight cancer, address climate change, cut out HIV, but it can also create anti-malaria mosquitoes and all sorts of things. This morning, maybe some of you heard, around 6 o'clock in the morning on NPR, right? A story about a woman who had just been treated with this technology, a clinical trial for sickle cell anemia. How close to altering the code of life are we?
So actually, scientists every day alter the code of life for research purposes. So this is what we do every day as biologists. Now what is really new with this technology is that prior to CRISPR-Cas9 and prior to the two other types of technologies that were used prior to CRISPR-Cas9 is the ability to really do so in human cells. And so this was really started with the technology called zinc finger nucleases and talon nucleases. So the ability really to reach the DNA of human cells and precisely alter the genetic code. And this is essential to understand the functions of genes. One has to really work with the multiple types of cells that are composing our body. And so that is really, critical. With regard to sickle cell anemia, this goes along with blood disorders that have genetic cause. So these are really disorders that are caused by a mutation in one gene. And where CRISPR-Cas9 is really important is that you have about 10,000 genetic disorders that are caused by the mutation of one gene, and maybe for 95% of them, there is no way to really cure the diseases. And this is where CRISPR-Cas9 can bring something. So for sure, it's difficult to say that it will be able to cure every types of disease, because it's a lot about being able to target the cells that are affected. But in the case of sickle cell anemia, it's a combination of what we call genome editing and cell therapy. So it's about taking the cells, the sick cells of the patient, bringing them into the lab, modifying them to rescue the wild-type phenotype and re-injecting them back to the patients.
And why start with sickle? Why was sickle cell this early?
But because actually this affects a number of people.
Right.
And also because it belongs to the cells that, it belongs to the disease that target blood cells. So blood cells are easily reachable. And also the type of mutation was a mutation, I'm not going to enter into the genetics, but in terms of the modification of the gene, CRISPR-Cas was in a way an attractive tool to be able to correct specifically this mutation.
And when and how will we know whether this story we heard today, this woman today, is cured or has a...
It will take some years.
Years, just of watching her and...
Exactly.
And so what will we be watching for? What will be...
Actually, the development of... actually the non-development of the disease in principle, and whether she's fine.
So one of the things that these pigeonhole questions allow me to do is to weave your questions in, and so there's a good follow question from Dan, which happens to have the most votes in the room tonight, which is today's announcement of this first U.S. clinical trial using CRISPR to treat this genetic disease, sickle cell anemia, gives hope to many. But what other similar CRISPR treatments seem close at hand? So what might be next?
So as far as I'm concerned, so cancer immunotherapy. But in this case, it's not, CRISPR is just a technology used to engineer immune cells. So the basis of cancer immunotherapy is to engineer our own, the immune cells of the patient in the way that those immune cells, once injected into the patients, can recognize the cancer cells and kill them. So this strategy is already existing, where CRISPR-Cas brings an additional asset to the strategy, is that it's a technology that facilitates this engineering of immune cells that will recognize the cancer cells and kill them.
Would you talk about the malaria-resistant or anti-malaria, I'll call them anti-malaria mosquitoes. How does that happen?
So this is through what we call gene drive. So this is the ability to somehow change genes in a population that will be transmitted to the offspring. And with regard to malaria, this is to engineer mosquitoes so that they cannot transmit any longer malaria. So this is one application of the gene drive. It's
How
really
close are we to that?
Oh, it's already working in labs.
In labs?
so the
No,
questions-
but how about being deployed?
But that's a huge ethical question as well, is whether it is, how do you say, acceptable to do so in terms of protecting the environment and not changing too much the population of mosquitoes in the world.
So let's talk about that for a few minutes, because you raised the issue of ethical questions. 3 years ago, we sat here with Jennifer Doudna, and she did as well. There are ethical questions. You can invent farmed super animals, right? You can resurrect extinct species,
Yes.
maybe,
Maybe.
Maybe.
It will take some work, but
right? You can cultivate, be careful, Jurassic Park is out there, wait. But you know, potentially cultivate human organs in pigs or in other animals. Where, is there a clear line for you? How do you translate your ethical concerns in general into any kind of specifics?
so first of all, we work, us biologists, we work with strict regulations. We,
Sure.
we cannot do what we want in our labs. And there will be certain applications that will not be wise in any case or ethically, how do you say, wise to pursue. Actually, regulations are in place. Now to work in laboratories, now the issues are more, for example, with regard to, it's one example of the engineering of plant crops, for example. So when one uses CRISPR-Cas9 to engineer a new plant crop, one engineers a plant crop whereby at the end of the process of modification, one cannot really differentiate between a species engineered with CRISPR-Cas9 and a species that would have undergone natural mutations in principle. And so the technology is very useful, nevertheless, to create plant crops that are clean, in contrast to the technologies that were used prior to CRISPR-Cas9 to engineer plant crops. So there is a need to adapt the regulations to what CRISPR-Cas9 can do. And for example, the European authorities have decided recently that actually the regulations for the CRISPR-Cas9 engineered plant crops will be exactly the same than the regulations existing for plant crops engineered in using tools that are different from CRISPR-Cas9.
A GMO.
Exactly.
Right.
And
Europe is very opposed to that, right?
Europe is quite opposed to this in principle, indeed. And there is also the definition of GMO, that is a genetically modified organism, implying that the organism that has been genetically modified has been modified in a way that foreign DNA has been introduced into the genomes of the species. But with CRISPR-Cas9, one can produce a very clean species.
So might there be more receptivity to a CRISPR-modified crop than to a GMO-modified crop?
Not specifically. I mean, for sure, us geneticists, if we modify the genome, we modify the genome. But at the end of the process, it's more what will be modified rather than the way
Right.
to modify the plants. And it's also, the responsibility to nevertheless consider that in light of the climate change, it will be also important to engineer plant crops that can survive the challenge of
Right.
of climate change.
This is the core of the ethical argument around GMO, right? We
Exactly.
can have a maize, corn, that can grow in drought conditions, or we can put more healthful nutritional ingredients into sorghum, so that can help prevent stunting.
Yeah.
But there's a great deal of opposition in some corners to that.
Yes, but there is also, I think, always a confusion of GMOs and the lobbying
Yeah.
behind GMOs. Because in a way, in the future, one could also think that it will not be possible any longer to figure out whether a species that is put on the market actually has been modified or not. And one could also think that it could actually promote the production, the local production of plants by farmers, plants that will just be more amenable for the conditions that are important for growing plants at a certain location.
So with questions like this, Linda asks a question that's related to this, which is CRISPR genome editing is amazing, she says, but how do policymakers prevent abuses of this incredible tool? So talk a little bit about how policymakers put the parameters around your work, perhaps, or help to define the ethical bounds of what you're doing, where the regulations are. How does this get done?
Yeah, so that is a little bit the concern of scientists.
It's concern of scientists? Okay, it's
Yes,
concern.
because actually that is what really we observe with CRISPR. It's a technology that goes very fast, and regulations that are not adapted to the fast evolution of technologies, but it's not only for CRISPR. CRISPR-Cas is a very good example of it. But this is always the redundant aspect of regulations that have not considered technologies that will be developing very fast and are lagging behind. And then policymakers that are not always well informed, because also the scientists are not, don't have the time to inform well the policymakers, and also because you have systems missing to allow communication between the policymakers and the scientists. And so this, I think, it's really an effort that has to be done in the future, because technologies will develop faster and faster, not only in biology, but in other fields. And so that is, in a way, a fascinating aspect. But this is also frustrating, because in principle, we have now technologies that can really be really impactful, specifically for plants. I mean, the production of plants that allow to feed the planet, and yet it does not go, fast enough to react.
Yeah, so this is moving very fast. It's very. Do policymakers even get it? Do they understand? Careful what you say now. We're in Washington, so.
Yes and no. I think they understand the generality, but they have difficulties to appreciate what is different with CRISPR-Cas9 compared to other gene technologies that were used prior to CRISPR-Cas9.
That frustrate you that they have trouble with that? I mean, is that?
Yeah, in a way, yeah, for sure. It frustrates scientists, indeed. Mm-hmm.
Well, good luck. Maybe you'll do better than the rest of us.
Yes.
But
we are used to be frustrated, because
exactly.
in general, nothing
true.
is working in science.
It's just another experiment. So ExxonMobil announced some time ago that they had used CRISPR to double the amount of biofuel that they were able to produce or generate from marine algae. I mean, that's just an amazing thing.
Yes.
So, where do you, if you had to predict, where the big breakthrough, the application, is going to be, is that it? Is it in disease? I outlined these three areas. Where are you watching most closely?
Me, I watch more the disease.
The disease
applications, just because I study bacteria for the purpose of understanding how bacteria cause diseases in humans. Having said this, I just want to mention as well that one should not think that every type of cancer will be treatable by CRISPR-Cas9. I mean, the cancer immunotherapy strategy is adapted so far on a very minimal number of cancer. But again, if you look 50, 60 years ago, the development in medicine has been tremendous.
It's
so
unbelievable.
That's why a lot of let's say, strategies are expected to be developed with CRISPR-Cas9 or other types of gene editing technology that may come up in the future.
You say things are going very quickly. So 3 years ago when Jennifer Doudna was here with us, she said, I'm looking at my notes, you'll see clinical application within 10 years, possibly even approved therapies. That would be 7 years from now. Are we
Yeah.
ahead of schedule? Is it moving
I
faster
think with
than that?
regard, for sure, with regard to cancer immunotherapy and
Yeah.
the treatment of certain
Yeah.
genetic disorders, yes, there is a chance.
Can we ask a couple questions about you and where this all came from and this inspired scientist that we have with us?
Yes,
Okay.
indeed.
There's a question from Jamie who says, your scientific career has been very nomadic, as we heard in the introduction. You just like moving around, right?
Yeah.
Based on these experiences, what advice would you give to an early career researcher about adjusting to this lifestyle?
Yes, I will say that it's really an inherent part of being a scientist nowadays, is really the mobility. Having said this, as a typical French student at the end of the 80s and beginning 90s, I was more happy to stay in Paris, and I had understood very early on that one step would be to go to the US, but then would be to come back to France, like a lot of French scientists. So it was very, very difficult for me to do the first move. But then I did my first move in New York, and I became in love with New York very fast, and I really enjoyed actually doing research abroad. And I think I would always encourage the young fellows who want to do science, because it's not only doing science, it's also working in an international environment, working with scientists to bring you a lot, actually. And also having the possibility to experience different views of doing science, even though now we work surely in a globalized world. But it's really the enrichment to work with people, to have the possibility to challenge oneself. And I think to be a researcher and have actually this sense of mobility, for me, has helped me a lot to always reinvent myself.
Reinvent yourself.
In a way, yes.
Really?
So I was always a reborn Emmanuelle each time that I was in a new country. But it's also because when you go to a new environment that you do not know, it's very challenging, and then you're pushed to how do you say, to be in a risky setting that will for sure lead you to ask different questions and to be in a state of mind where you're more ready to take risks. And it's important to take risks.
I'd like to ask the audience, how many scientists are in the audience this evening? How many of you have moved around to different labs to pursue your research? How many of you found that difficult? How many of you found it inspiring? Was it inspiring or difficult for you?
For me, it was more inspiring than difficult.
You were going to where the resources were and to where people...
Yes, because at some point I was really, let's say, going where I found that the opportunity will be best for me to develop my career and my science.
So is this about funding? You spoke about funding earlier.
It's about funding, but I think ultimately, for sure, the career of a scientist is difficult, but you really also come to the point that scientists are also nice and that it's good to go to other countries. The people, to a certain extent, are very welcoming, and that the scientific community is quite international and welcoming as well. But the funding, surely, and also possibility to take a position. I mean, you don't have positions of assistant, associate, professorships opening every day, so. The market is also competitive, it's tough, and you need also to find the right institution that will allow you to do the science you want to do.
You and I spoke a bit about curiosity-driven science. We also spoke about funding. Are you concerned you've seen it both in American labs and in European labs, you've been all over. Are you concerned about the funding environment for curiosity-driven science?
For sure, because I mean, artists are always concerned, scientists are concerned, because if there is a funding that has to be cut, it will always be on the cost of actually art, culture, science.
Is it worse than it's been before?
No, I will not say that it has been worse, but the fact is that you have also more and more scientists. You have a lot of universities that have been created, lots of research institutions, so this requires sustainability of funding, and there is always a risk, nevertheless, that basic science is not properly funded. And in times of, let's say, of also of direction of politics that is also very much protectionist. We see this in Europe and also maybe in the US. Scientists are always fear that this direction of political views and may also affect science.
Sure.
So, because science is also a lot about how also, development also of, human, how do you say, potential, and it's always easy for certain political directions to make sure that maybe we don't do too much science, people become too much intellectuals, etc. So it's and also a fear of a world that is evolving fast with regard to technology, knowledge, innovations. So this it's important to maintain, and basic science is really at the origin of any types of innovation at the end of the day.
Yeah.
I mean, from the cell phone to even economy relies on mathematical models. And actually, even you heard this for Mr. Kavli, but a large number of entrepreneurs have also had an education in hardcore sciences, whether they continue in basic science or not. And basic science, it's important to provide really good training and education.
Let me come back to some of the questions to your science and to your research for a couple of minutes, because they're a good one. Jetta asks, many genetically related conditions are not coded in a single gene, but rather spread across many genes. How would CRISPR account for that, as well as the interaction aspect of gene editing?
Yes, so the CRISPR-Cas9 technology is well adaptable, at least not for research purposes. For research purposes, one can modify multiple genes at once to create modified model organisms. But with regard to, for example, treating diseases, the technology is adaptable for monogenetic diseases. For plurigenetic diseases, it's more complex.
There's another one here. This is anonymous, so we'll treat it such. CRISPR is part of the bacterial immune system. How unusual is this, is the question. Are there other research tools that can be developed from the genetic material in other species?
So CRISPR-Cas is not unusual in terms of thinking of a mechanism that bacteria and also archaea, other types of prokaryotes, have evolved to defend themselves against genome invaders. However, what was very, let's put it that way, enlightening for as a scientist is to see this very, very neat mechanism. So, as I said during my talk, CRISPR-Cas9 is only one mechanism of the multiple CRISPR-Cas mechanisms existing. What is fascinating is really how defense system has evolved in multiple mechanisms, employing very precisely proteins and RNA molecules to attack DNA in a very versatile fashion and multiple ways. So this is maybe for people who do not know so much about bacteria, something that is very, very, very surprising. I mean, bacteria are extremely small, but they are extremely powerful. We are made of bacteria. It was also mentioned that, the discovery that bacteria surely drive our our physiology.
Right.
And bacteria are everywhere in principle. And the fascinating things when you work on CRISPR is that it's about bacteria defending themselves against phages that are the most abundant population of organisms on the planet.
I'll ask one or two more from here, and then if you've got a question you'd like to step to the mic, why don't you do that? We'll have a few minutes to take some questions from the floor. Here's a question. Can you explain the difference between how CRISPR-Cas9 works and how traditional restriction enzymes work?
So restriction enzymes are really proteins so a family of proteins. Each protein can recognize a specific motif on the DNA. So one protein for a specific motif on the DNA. Like a software would recognize only one word in a text, one single word. The CRISPR-Cas9 technology can cleave DNA, but is a technology that is programmable. So you can program the technology to recognize different motifs and cleave different motifs of DNA. So this is a comparison between restriction enzymes and proteins. So these are both are cleaving DNA cleaving proteins, but CRISPR-Cas9 is programmable to be able to cleave the DNA at multiple positions.
That word programmable is so important. So let's start over here. Tell us who you are, and if you keep your question brief, we can get as many in as possible.
I'll try. So my name is Lamine Hendricks. I am not a scientist, so please bear with me. I'm still very fascinated by the how of CRISPR-Cas9 before I can even think about sort of the larger policy implications. You say that you program CRISPR-Cas9 to cleave genetic material, to delete, replace, modify. How does that programming take place? How do you program it to target a specific gene? And as a follow-up to that, how do you do so in a way that ensures it won't damage the organism that you send CRISPR-Cas9 into?
Yes. So CRISPR-Cas9, as I said, is two components. You have the protein Cas9 that is going to cleave the DNA, and the programmable component is of RNA nature. So it's a very small molecule that you can design to recognize DNA. So this is an RNA molecule that has a sequence that can base pair with the sequence of the DNA, and this base pairing brings Cas9 to the piece of DNA to be cleaved. So there are ways to design the molecule such that the RNA molecule will not lead to what we call off-target effects, meaning the protein recognizing another site of DNA than the site to be cleaved. So the technology has evolved, and there are certain rules how to design the technology. Now, with regard to the modification of the genome or integrity of the cells, it's less about CRISPR-Cas9 than the delivery technologies that will bring CRISPR-Cas9 to the cell. For sure, each time that we introduce a gene editing technology into a cell, we use a delivery tool that affect the cells. But in principle, now you have delivery technologies that are also well developed to be specific with regard to being targeted, to target the cells to be modified by CRISPR-Cas9.
Yes, sir.
Thank you, and thank you for talking to us tonight, and congratulations on your work. You talked about regulation. Specifically, you talked a lot about the regulation in the framework of the European Union and some of the more restrictive regulations there. It's possible to imagine societies where things maybe aren't quite so restrictive, and I know there was some controversial work last year coming out of China, for example. Yeah. I'm wondering if any sort of larger international structures exist governing this technology. If not, should there be? And if there should, what sort of form you think those kinds of structures should take?
Yeah. So the CRISPR babies.
These were twin girls, right?
Exactly.
From China.
That for sure happened in China. So this has been surely criticized by the scientific community, and not only the scientific community. So these types of experiments, for example, will not be possible in Europe, where there are very strict regulations that forbid to use the technology for human germline editing. The technology is possible in certain countries to modify the genomes of embryonic human cells as long as it is for research purposes and as long as the cells are actually the cells used originate from cells that will be discarded otherwise and issued from in vitro fertilization. So this type of research is not possible in a large number of countries, first of all, and second of all, the CRISPR-Cas9 technology is not yet here in any case to modify human germline without, let's say, implications on the human genome of the cells, of the human germline cells that are modified by CRISPR-Cas9.
But the point is, there is no international, globally accepted authority or set of regulations that is going to govern this in a uniform way worldwide at this point.
No, and it's difficult to imagine that everyone will come.
Does
to
that
the
bother
same.
you? Does that concern you?
Indeed.
Europe can have as wonderful regulations as it wants. If China or somebody else wants to go in another direction.
Indeed. But this is as such. So the only thing that we can hope for is that discussions are continuing to be able to really allow the public to understand the implication of and the danger of the editing of human germline.
Is there something more the international scientific community can do, do you think, to influence that? That's a huge question, right?
I think this is also in the hands of the policymakers in principle.
Sure. Yes, ma'am.
So we often hear about CRISPR being cheap and easy. And I get the cheap part, particularly from the perspective of the medical researcher. But I look at all these companies that have popped up around this. If one day I need to benefit from the applied therapy that comes out of CRISPR, do you think I'll still be using the word cheap?
So it's a very good comment indeed. So if you really take into consideration the technologies that we are using in labs, for certain questions that we want to answer, we have to use equipment that is quite costly and which not every lab can have access to. With regard to technology that is needed every day to modify the DNA to be able to understand the functions of genes, CRISPR-Cas is a technology that any lab can employ because it's affordable. With regard to the costs of treatment, I cannot comment on it, but surely, because I'm not a specialist, but surely this is also a question that is extremely valid. It's nice to have a technology that can, and this is not only for CRISPR-Cas, for other types of technologies. It's nice when we have a technology that opens doors for the treatments of diseases that could not be treated. But the thing is that who will have access to the treatment? And this is where it becomes even more complicated because you need to have a system in place to be able to provide the patients the means to be treated.
We'll figure that out here in this country.
Yes. Yes.
All right, I think
Indeed.
we have time. We're almost out of time, so we'll have time for two more from the floor. I'm sorry to the others, but you may be able to hover. Go ahead, ma'am. Yes, please.
My name is Mary Fraker, and I am just a non-scientist. My question is about sickle cell, and it's my understanding that the mutation or mistake that causes sickle cell also conveys immunity or at least resistance to malaria. And if that's true, I'm wondering if the precision of the work that you're doing would allow that resistance to malaria to remain while curing the sickle cell.
So this, I cannot comment on this aspect. I don't think, in any case, the way the technology is used for the treatment of sickle cell anemia is, for what I know about the way that it is done, has nothing to see with the fact that the patient will become more resistant to malaria. And the fact is that the patient is more interested, first of all, to be treated for the disease.
Yes, sir.
Hello, my name is Mike, and I'm a neuroscientist, but more of a dreamer. So I think symbolically with the last question here, I'm thinking long-term possibilities with CRISPR to the point where we're inhabiting other celestial bodies, and where are we now? So my question is related to the digestive system. We talked some tonight about crop engineering, and you mentioned the word human physiology, and we can alter this. Supposing, again, pardon me, but thinking several hundred years from now, 1,000 years from now, and we get to the point where we can't engineer the crop, so we have to go to plan B, engineer our digestive system. Will CRISPR be able to do that a long time from now, and therefore every human born thereafter can have a new digestive system, and now we've really altered what we are as humans?
You mean altering the digestive system
So we
through...
can get nutrients from that which we now would otherwise not consider food, or things that plants that...
No, I think this will be a bit science fiction, so I don't think that we will reach this point. And I think as a French, I can also, I mean, I enjoy eating. And I just want to, I just think that human beings should continue to enjoy the real food.
I think you have a conflict of interest on
exactly,
this.
I have a conflict of interest.
One more from the floor here, which is in a 2016 profile, you told Nature you have a few non-CRISPR things up your sleeve. Can you tell us about what's on the horizon for you?
Yes, so in the lab, actually, we work on different mechanisms that are actually like CRISPR-Cas using proteins and RNA molecules, and mechanisms that allow bacteria to defend themselves against different invaders like CRISPR, but also allowing to the understanding of how bacteria can survive in the human host and can adapt to the human host. So we also have some research projects that are the interface between the interaction between bacteria and human immune cells. So it's difficult to enter into the details, but I do hope that at least certain lines of research will lead to new anti-infective strategies. So this is the reason why I do research. But you know, for CRISPR, it took me maybe 20 years. So I think right now I'm very busy in actually promoting also CRISPR research. But I give myself another 20 years to hopefully discover a mechanism that will have also a large implication, specifically for the treatment of bacterial infections.
That would be huge. Well, we help give you those 20 years and many more. We'll end where we started. You said, I'm just a scientist. No, you're not. You're much more than that. I know that. You're right? You're a sportswoman as well.
Yeah, I became recently, in principle.
Okay, so just tell people what sports you're into.
I'm into running, biking, swimming, functional training, boxing.
Boxing. I want to thank you on behalf of everyone who's here.
Thank you very much.
Your work with CRISPR has been remarkable. It holds such excitement, but also some very difficult discussions and decisions for policymakers and others around the world. But it's enormously exciting. It shows what science can do, the contributions that you and scientists make. So thanks to you, thanks to Kavli, thanks to Norwegians. Thank you very much for being here.